Then we have attached adapters to the ends of these fragments. So why we attach these adapters? This process is basically enabling them to efficiently amplify and sequence our DNA that we have extracted from our sample. So after library preparation, we'll go for the sequencing. So sequencing approaches that we were discussing about Illumina sequencing, Pyrosequencing, Selecta technology, and ion torrent. So these are the different approaches. There is minor differences in these approach. What that difference? The first one is pyrosequencing. So library after DNA fragmentation and adapter attachment, we those will attach those DNA fragment, and you can say we will introduce those DNA fragments with adapter ligation onto flow cell. Here you can see in this picture the one diagram, the one term is bridge PCR. Can you see the bridge PCR and immersion PCR? So डीएनए फ्रेगमेंटेशन के बाद क्या करते हैं हम कि उसको हम फ्लो सेल पे इंट्रोड्यूस करवाते हैं। फ्लो सेल पे जो हमारे डीएनए फ्रेगमेंट्स हैं विद एडॉप्टर, दे विल अटैच एंड they will bind to the complementary beads in emulsion case, and in bridge PCR they will attach with the complementary adapter. So complementary adapters are the complementary sequence of those adapters that we have attached with our fragments. So that are the nucleotide sequence that is complementary to the adapter, and adapter is attached to DNA fragment. So after bridge PCR and immersion PCR, in immersion PCR we'll attach these DNA fragments with adapter with oil beads. So immersion PCR will attach beads with these adapters. In bridge PCR we'll introduce our DNA fragments with adapters on flow cell. That flow cell have complementary nucleotide sequence. So after that, how we can find out and how we can determine the sequence of DNA. So if we talk about pyrosequencing, uh, you all know very well DNA is the double helical structure. So we have fragmented our DNA. We have shared our DNA. After sharing, we have single fragments. So so how we can sorry so how we can analyze our desired sequence. So when we'll introduce our DNA fragments on bridge PCR, either on emulsion case, attachment of beads. So we were discussing about pyrosequencing. So in pyrosequencing, when our complementary nucleotide, that is DNTP dinucleotide, that can be adenine, that can be guanine, cytosine, thymine, whatever is the complementary of our template DNA, when that DNTP will attach, will bind to our template, our DNA fragment, light will illuminate in case of pyrosequencing. So that light, so we will detect that illumination of light by using detector. So for example, we have four DNTPs A, T, G, C. You all know very well adenine, guanine, cytosine, and thymine. So when adenine will attach, will bind to the complementary sequence. We have attached adenine with blue color with blue dyes, guanine with red dyes, cytosine with yellow. So in such a way, we have attached different dyes with different DNTPs. When complementary nucleotide will bind to the will bind to their complementary nucleotide, light will illuminate and we will detect that light, and that light will generate peaks in different colors. So we will detect, we will attach our sequencer with computer, and that computer will analyze those illumination of light, and those illumination of different peaks will generate, and we can in such a way we can analyze our whole DNA sequence. The next that is Flexa technology and Illumina technology. How that technology is different from pyrosequencing. Pyrosequencing, we know. We were next slide, please. We were discussing about pyrosequencing. Light will illuminate. So how that light illuminate when whenever our complementary DNTP will attach pyrophosphate will release pyrophosphate. That pyrophosphate will generate light, and that light is used to detect the sequence of DNA. And the next method that was the so and the next method and the next approach, that is here is also pyrosequencing. We have discussed very well extraction of DNA, fragmentation, library preparation, and then we'll go for the then we'll attach with the beads, and after that we'll introduce complementary DNTPs. When complementary DNTPs will attach, pyrophosphate will release light, and we'll detect that light signals, and will generate peaks, and their different peaks will help to analyze whole DNA sequence. And the next approach that is different from the pyrosequencing, this figure is also about pyrosequencing, how light generate. So basically, when DNTP is attached, complementary DNTP attached, pyrophosphate will release. Well, when pyrophosphate release from the attachment of DNTPs, that will generate ATP, and generation of ATP basically release light, and that light is used to detect our sequence of DNA. So this figure is also explaining about the phenomena of pyrosequencing approach. And in Flexa technology, what's the difference and how the technology is different from pyrosequencing? In Flexa technology, we introduce after the attachment of adapters, we'll introduce on the flow cell, on flow chip. You can say chip on chip, when you will introduce your DNA fragments with the attachment of adapters that will bind with the like complementary adapters will ligate with the adapters. So they will generate same, just like pyrosequencing, the pyrophosphate release, and that pyrophosphate release of pyrophosphate will generate ATP. But in case of Flexa technology, there is no release of pyrophosphate. Only you will introduce on chip, and your chip has thousands of DNTPs that are labeled with different colors. You have used four dyes that are used to differentiate your four bases DNTPs, A, T, G, C. So we know very well our whole DNA has a repetition of thousands of A, T, G, C. So we'll just use four dyes and label the four DNTPs, and we'll introduce thousands of DNTPs labeled with four dyes on chip. And when we'll introduce our DNA attached with adapters on that chip, that will bind with the complementary adapters. When that will bind with the complementary adapters and with complementary DNTPs, that will light will generate, different peaks will generate. So your detector will capture those peaks, and you can analyze your sequence of your DNA. Next slide. So here are the applications that we have already discussed. We can use here we can use next generation sequencing. We can use next generation sequencing for different disease, for different research purpose, for the study of different disease, for mutation analysis, for quantification, for the treatment of different for the treatment of different disease. We can use next generation sequencing approach. And next topic, according to your content, that is central dogma as per my content. Before going through next generation sequencing, you should have knowledge about structure of DNA and central dogma. Uh, but according to your content, next generation, you have studied next generation sequencing. Now we are going to study central dogma, although these two topics are interconnected. Now these two topics are different. In next generation sequencing, basically we study sequence of DNA. So what is DNA and what is the structure of DNA that we are going to study in central dogma. So what is central dogma? Central dogma basically central dogma basically is the flow of gene information. So we can say a flow of gene information that is from one generation to next generation, and within one generation flow of gene information that is called central dogma. And in central dogma, three processes are involved. DNA to DNA formation, that process is called replication. DNA to RNA formation, that is called transcription. And messenger RNA to protein formation, that is called translation. So these three processes basically are the central dogma of molecular biology, and these three processes, replication, transcription, and translation, are involved in the transfer of in transfer of information from one generation within one generation and from one generation to another generation. So here we are going to study about structure of DNA, DNA replication, transcription, and translation. What is DNA and what's the structure of DNA? Here are the scientists who have discovered structure of DNA. Watson Crick was the first who discovered the structure. So what is DNA? DNA is the heavy material, is the you can say is a genome that is involved, and that that DNA and that genome is involved in the formation of protein. And I think you all know very well proteins are enzymes, and enzymes are used in the regulation of metabolism. So DNA is involved basically in the formation of protein. So DNA, what is DNA? DNA is a single large molecule that is double helical structure. Double helical structure comprised of three different components. Next slide, please. So, what are the different components that are involved in the formation of DNA structure and that are involved in the structure of DNA? Sugar, pentose sugar, that is called ribose. Four types of bases are involved in the formation of DNA, that is adenine, guanine, cytosine, and thymine. So basically, whole strand of DNA comprises up to three components. Here you can see bicarbonate sugar, that is pentose sugar, that is ribose sugar in case of RNA, and in case of DNA, that sugar is deoxyribose. So why we call that ribose DNA? Because on second carbon of that pentose sugar, hydroxyl group is absent. So that's why due to absence of hydroxyl group on second carbon of that pentose sugar in case of DNA, that sugar is called deoxyribose, and in case of RNA that sugar is called ribose. And phosphate group is attached on the fifth carbon of pentose sugar. And two ends are joined through hydrogen bonding. So basically hydrogen bond is involved between bases, between nitrogen DNA strands. And as we discussed, four types of bases are involved in the formation of DNA structure A, T, G, C. Adenine will always pair with thymine, and thymine will always pair and will hydrogen bond with adenine. And guanine will also will always pair with cytosine, and cytosine will always pair with guanine. Two hydrogen bonds are involved between adenine and thymine bonding, and three hydrogen bonds are involved in guanine and cytosine bonding. In this figure and in this structure, you can see where adenine combined with thymine, only single hydrogen bond is involved. Whereas in guanine and cytosine, two hydrogen bonds are involved. If we talk about single strand of DNA, although both strands of DNA comprises up to pentose sugar, that is deoxyribose sugar plus nitrogenous base is attached on first carbon, phosphate group is attached on fifth carbon, but the single strand, two strands connected with connected through hydrogen bond with one another. If we talk about single strand of DNA, how these three components that are repeatedly present in single strand, how these three components join, pair with each other with one another, they pair single strands. In single strand, these three components, phosphate, pentose sugar, nitrogenous base is involved pairing section with the phosphate group that is attached to that carbon. That phosphate group bonded with the third carbon, hydroxyl group. So hydroxyl group is present on the third carbon. So third carbon and fifth carbon coordination is involved in the joining of these three components in single strand, and the bond that is present in single strand between phosphate and hydroxyl group between fifth and third carbon, that is joining of fifth and carbon, that is phosphodiester bond, and the bond that is involved in the pairing of two strands, that is hydrogen bond. So in single strand, phosphodiester bond is involved in the backbone in the joining of these three components. Next slide, please. So that was the structure of DNA. Here we are going to study about DNA replication. So what is DNA replication? The process of formation formation of replica from one DNA. You can say generation of copy of DNA. The process we generate copy of one copy of DNA. That process is called DNA replication. So DNA replication, in DNA replication we generate copy of DNA from DNA. How the copy, how we'll generate that copy in DNA replication? So as we discussed in structure of DNA is double helical structure, comprises of two strands that are interconnected with each other through hydrogen bonds. So what will happen when we are going to copy that strand? Enzyme will will cut hydrogen bond that is involved between two strands. So enzyme will cut the hydrogen bond and will unbind. And when will unbind, after unbinding, after cutting the hydrogen bond tearing, that is involved in the tearing and connecting one strand with another strand, we we can obtain two strands. Here in this structure, you can see we have cut the hydrogen bond and we have separate out two strands, unbind strands from one another. So in next step, when we cut hydrogen bond, we will obtain two single strand. So when we unbind two single strand, you can see here is the fork-like structure. Fork-like structure will generate when we will unbind two strands and when we'll separate our two strands from one another. So after unbinding, after separating these two strands from one another, what's next? We'll start replication. Previous slide, please. So we'll start formation of second strand of these two single strands. Because we know DNA is a double-stranded, double-helix structure. So when we will unwind, we will obtain three two single strands. Now we have to synthesize second leg of that single strand. And replication always starts from 5' to 3' direction. So here you can see in one strand continuously enzyme will unwind, break the will break hydrogen bond, will unwind, and the DNA primase will attach DNA primase. Then DNA polymerase will come. DNA polymerase will start synthesizing new strand. How that will synthesize? DNA polymerase will attach complementary bases. For example, our template. What is template? Template basically is our unwinded strand that we have separate out from one another. That you can say that is parental strand. So DNA polymerase will attach complementary bases to the template. So for example, our template strand have A. So DNA polymerase will attach A on new strand. Template has G. DNA polymerase will attach C. Template have C. DNA polymerase will attach G. Template have T. DNA polymerase will add A on new strand. So in such a way, DNA polymerase continuously synthesize a new strand. Our one DNA is complete. But in next strand, you can see uh, that is lagging strand. Why that is lagging strand? Because in one strand, replication start from the 5' to 3' direction on 3'. If you see in this diagram, your next strand is 5 and the replication will start from 5'. Means replication will start from the opposite end because that 5' strand have their opposite 3' end. So replication always start from the 5' to 3' direction. So this strand on this strand will start synthesizing DNA whereas in opposite end your unbinding of strand will start in opposite direction. So in this way, DNA polymerase will attach complementary bases in opposite direction, and your strand is unbinding in opposite direction. So you can in on that strand that is lagging strand, you have obtained different patches. You have synthesized new strand in patch-like structure, and that patches are called okazaki fragments. After synthesizing new strand on lagging strand, DNA polymerase will come, that will proofreading, that will fill the gap after adding and after introducing complementary bases and ligase. Then ligase will come and ligase will join those patches with one another in such a way we can obtain from one DNA two DNAs in DNA replication. Next slide, please. So we were discussing about central dogma that is involved in the genetic continuity, either that is within one generation, either that is from one generation to the next generation. Two processes are involved in central dogma. The first one is replication that we have discussed. Genetic information is transferred from DNA to DNA. From one DNA, we can obtain two DNAs. We can prepare two DNAs, two copies from one DNA by using replication process. And the next process that is transcription and translation. DNA to protein formation is called central dogma. And transcription and translation are involved in protein formation. How transcription and translation are involved and what's the difference between transcription and translation? Basically, in transcription, DNA strand is transcribed into messenger RNA. And that messenger RNA basically copies the information of DNA and used to for the protein formation. So formation of DNA to RNA is called transcription. So in transcription, only one of the we know very well DNA is double helical structure, two strands are involved in the formation of DNA. But in transcription, only one strand is synthesized from only one of the two strands of DNA. And that single stranded messenger RNA is transcribed from DNA. That is called transcription. Here you can see RNA polymerase identified the coding strand from one of the two DNA strands. After finding coding sequence, coding strand, RNA polymerase cut the break down hydrogen bond from that part, from that region, and start synthesizing complementary messenger RNA. So RNA polymerase transcribe the DNA information into messenger RNA formation. But the difference is that we have studied in structure of DNA, four types of bases are involved, A, T, G, C. The difference between DNA and RNA structure is that four types of bases are also involved in RNA formation, but thymine is absent. Instead of T and instead of thymine, guanine site is near adenine. Instead of T, U is present in messenger RNA. So we in you can see T always paired with A, and A is the complementary of T. But when we are going to transcribe DNA into RNA, in RNA, U is the complementary of T. You can see here. So four types of bases are also involved in the formation of RNA and RNA. The difference between bases is that instead of TN and instead of thymine, U is present in RNA. Next slide, please. So when does transcription start or stop? We have discussed where RNA polymerase will find the coding sequence. RNA polymerase will start transcription. When RNA polymerase will find non-coding sequence, it will stop messenger RNA transcription. It will stop transcription. When transcription will stop, translation will also stop. And translation will stop means proteins will not synthesize when transcription is stopped. So basically, whenever when needed, turn on and turn off. Next slide, please. So here is the last step of central dogma translation. So that messenger RNA that we have transcribed from DNA, that messenger RNA used to translate protein. So formation of protein from RNA is called translation. Just like transcription, translation, when translation will start and when translation will stop. Here, just like transcription in translation case, also start codon and stop signals are involved in the formation of protein. When transfer protein will find out start codon, start signal the formation of protein, and when it will find out the stop signal, it will stop the formation of protein. So that's all about today's lecture. If you have any question, you can discuss.
Is this the last slide? Last slide?
Yes, it's.
Yeah. Any question?
I have
Any
already
question?
discussed here that native codon, start and stop codon that are involved in the protein on and off regulation.
Any question from the students can raise their question. Ma'am is still here. She will address the questions. It was a good lecture. We learned a lot. Thank you so much, ma'am, for your availability. Uh, Students, any question, please ask. Sir Ghulam Murtaza, where are you? Sir, sir, आ गए लेक्चर एंड हो गया
Students, if you have any question, you can discuss. We can discuss.
लेक्चर एंड हो गया Any question? Can ask anyone if have any question? Can ask.
Yes. Am I audible, ma'am? Ma'am, can you listen me? Ma'am, Ammara, can okay, thank you so much. Okay, thank you so much, everyone, for joining today's session. And thank you so much, Ms. Kumar, for insightful question on next generation thinking and centered over. Yeah. So if anyone have any question, you can ask.